Dear students, today I am going to deliver a lecture on the topic molecular marker. This topic has become very much pertinent in modern plant breeding, and it is also very much important for your different competitive examinations in plant science particularly, which takes a lot of questions from this part, molecular markers. And it is also pertinent not only for agricultural sectors but also for other examinations like CSIR, GET and so on. So the first, I will be showing the breakup of this lecture, which contains mainly 4 parts. The first part is the general structure of the plant genome. Without understanding this, you can really not understand how the molecular markers can actually work. Next is the general structure of the plant genes, and third I will be discussing about the 10 basic type of molecular markers starting from RAPD to T-arms, and I will be giving you some of the readings for reading in your further, who are interested in further knowledge in this aspect. So if we look at the general structure of the plant genome, it is defined as the complete set of chromosomes or genes, or it is the sum of all genes and intergenic regions. So these intergenic regions are regions in between the genes that code for the proteins, but we previously thought that probably these intergenic regions are not having some genetic information. But regularly our understanding about the gene is changing, and we know today that these intergenic regions are also very much important for regulating several gene expressions. So when we call up the plant genome, it's basically containing 3 compartments. One genome, we know that the nuclear genome, where the chromosomes are present, and there are 2 more genomes, that is plastid genome and mitochondrial genome. The animal cells, they generally contain only the mitochondrial genome, but the plant cell has both the genomes, plastid and mitochondrial. So when we talk about genomic DNA, it is actually a mix of plastid genome, nuclear genome as well as mitochondrial genome. So if we try to find out the features of the nuclear genome, you will find that it is mainly composed of non-coding repetitive DNA, and only a small fraction of the genome codes for the proteins, and it contains 2 fractions. The genomic DNA contains 2 fractions. One is the genic region and the non-genic region, that was I was telling just a few minutes back. The genic part contains single or multi-copy genes and tandemly repeated genes. These are the genes which code for the proteins, and that somehow code some enzymes to regulate several biochemical pathways. And then the non-genic part contains tandem and dispersed repeats, mainly transposable elements. In other lecture I will be discussing about these transposable elements. So there are 2 main types of transposable elements. One is transposon and the second one is retrotransposons. And retrotransposons actually replicate within the genome via an RNA intermediate. So the non-genic region is actually not non-functional, that I was highlighting. There are several regulatory elements that are present in this non-genic region, and the DNA content in a haploid cell may not reflect its genome complexity. So this is another topic that needs a separate lecture on C-value paradox, but for today's understanding it's not very much important to understand the C-value paradox. Next, if we go for the plastid genome, you will see that it is generally maternally inherited. It contains circular double-stranded DNA. Each leaf cell contains around 50 to 200 copies of the plastid genome, and it codes for around 140 genes, and the products of which are mainly necessary for the process of photosynthesis. So it has a separate gene expression machinery also. As you know that plastid and mitochondria were once the free-living bacteria, and the endosymbiotic theory through which they have been incorporated in the plant cell and became the integral part of the plant genome. So the replication and its division is also independent, and the size of the genome is very small, around 270 kb, and plastid genome is almost similar in different plants. So now if we look at the mitochondrial genome, you will find that it is also maternally inherited. It contains circular double-stranded DNA. A part of the genome may exist as a separate subgenome within mitochondria, and it codes less number of genes, generally less than 100 genes are coded by the mitochondrial genome. Separate gene expression machinery is there. It's also having its own replication and division, and the size of the genome is quite bigger than the plastid genome. It's around 2500 kb. So whenever we talk about any character and its phenotypic expression, so that is basically an interplay between the genes present in the nuclear genome, the plastid genome and the mitochondrial genome. So here I have shown some of the chromosome number and genome sizes of different crops. Here you can understand that the complexity of the genome or the number of genes it is coding and the amount of expected genes, it's not having a linear equation. For example, rice is containing 24 chromosomes with a genome size of around 430 megabase, and the expected number of genes is around 40,000. But if you see here that the pigeon pea genome, it's containing 22 chromosomes, but the genome size is almost double of rice, around 850 MB, but coding for relatively less number of genes, that is around 50,000 genes. In case of tomato also, you can see there are 24 chromosomes having almost more than double size of the rice genome, but the expected number of genes is less than the expected number of genes in case of the rice genome. So this is a general structure of the plant gene. As you have studied, that a gene is under a transcriptional regulator which is known as the promoter. Then you have, in case of eukaryotic genes, these are actually split genes that contains exons and introns. The exons are the protein coding parts, and the introns are generally removed from the exon through the process of splicing, and this splicing makes the mature mRNA which is coded to a protein, and that very protein is translated, and that very protein comes into action through a mode of enzymatic reactions in several biochemical pathways. And it also has a 3' UTR region. So both the promoter and 3' UTR region, they are not coding for any kind of protein, but the expression of the gene is basically controlled by these two elements, and sometimes the intronic regions present in them. So here I have shown a structure of a gene which has a 5' UTR, untranslated but transcribed region. There are exons named as E1, E2, E3, and there are two introns, I1 and I2. There is 3' UTR, and there is terminator named as T. So first let's look that how some changes in this element can actually change the function of the gene, right? So mutation, if takes place in the promoter region, then what will happen? This mutation will lead to transcriptional deregulation, means this is the region where RNA polymerase comes and binds to have the transcription. But if some mutation occurs in the promoter region, then the expression of the gene in terms of RNA is going to be suffered. Now, if mutation takes place in the 5' UTR region, then maybe some post-transcriptional regulation of the gene. If some mutation takes place in the exonic regions, then they are going to be very much detrimental. You know that exons are the part which contains the codons, and if here some changes take place, then the codon will be changed, and the respective protein will be having a change in the amino acid sequence also. So mutation in the intronic regions may also lead to altered function, because you know that the proper removal of the introns from the exon is a sequence dependent manner, and if the sequences in the splicing donor and acceptor sites are changed, then some introns may remain in the transcript, which can go for alternate splicing and non-canonical splicing. So these are the regions where mutation in the 3' UTR again can go for some post-transcriptional deregulations. So mutations are the source of the sequence variation that I was talking about. Now if we try to define mutation, it is a sudden heritable change, and it can be spontaneous or induced. So induced mutagenesis is again a different topic that can be dealt in a separate lecture. And let me define some of the basic type of mutation. So point mutation means it is a change of the nucleotide sequence. It may be a substitution, it may be an addition or a deletion. So purines, when purines, that is A and G, are replaced by purine itself, then we call it a transition mutation. And when purine is replaced by pyrimidine or vice versa, then we call it a transversion, right? So point mutation not changing the amino acid. If some point mutation has taken place, but the corresponding codon is coding for the same amino acid, there is no change in the amino acid level, then we call this mutation as the silent mutation. So if a point mutation changes the amino acid, the sense of the amino acid is changed, so we call it a missense mutation. And if a point mutation introduces a stop codon, then we call this mutation as a nonsense mutation. So these type of mutations, when they take place in the gene, then the function of the gene is altered. And when we talk about the markers, our basic target is to identify this kind of mutations, which can actually cause the change in the DNA function, and we can predict the phenotype even by analyzing the DNA only. We need not to wait for the phenotypic expression. So insertion or deletion of number of nucleotides that is not divisible by 3 in the coding DNA sequence causes a shift in the open reading frame, and this mutation is known as open reading frame mutation or frame shift mutation. So here I have given an example that suppose this was the initial sequence and I have written these sequences in a triplet manner 333. So suppose the T is omitted, T is deleted, then the reading frame changes like GGA, GCT, then CGC, CAG, and you look, here comes a new TAG codon which has not come due to any insertion or any deletion, but rather the open reading frame lost the T deletion, so the triplet reading frame is now changed, so there is an initiation of or incorporation of a premature stop codon. So often this kind of frame shift mutation, they introduce some premature stop codons. But if the number of bases which is inserted or deleted is divisible by 3, then what happens? Then the particular part that is inserted or deleted may have extra amino acid or reduced number of amino acid, but the reading frame doesn't change, right? Because always reading frame is read in a triplet manner. So here I have given another example where TG is deleted, and you can see that again a premature stop codon TAG has come from the same sequence which was not having the TAG. So now I come to the molecular markers with this basic understanding of the plant genome and the plant genes. So what the molecular markers are? They are basically the DNA sequence features used for mapping, identification or introgression of a trait through genotyping. Simply saying, a marker is a kind of molecular device which gives you the power to predict a phenotype even before the time. For example, suppose you know that a particular gene A is controlling the fruit color in tomato to be red, and it's known to you that a deletion in A gene causes a yellow fruit, right? So if you are given a seedling and if you want to predict that which color of tomato this seedling is going to bear when it is becoming a complete plant, then you will have to wait for flowering and initiation of fruiting and maturity of fruits. But if this DNA sequence information is available with you, you can run a PCR to know that whether this seedling is having a deleted sequence or a normal sequence. If you find by analysis that it is containing a deleted sequence, then you are 100% correct in prediction that this line is going to be a yellow fruited line. Or if it is not having any deletion, you can say that it can bear the red fruited line. So this is the basic purpose of using a molecular marker. So I have given a very simple example. The complex examples may be identification of disease resistance, identification of nematode resistance, and so on. There are several traits which are difficult to observe. Some traits are underground traits like root morphology. So it's not possible to every time go and dig up the plant in a non-destructive manner to score it. But through the DNA marker technology you can have it. And DNA based markers can be either dominant or codominant. So here also the dominance and codominance term is just like the Mendelian phenomena. You know that a codominance gene shows its heterozygosity, because suppose I cross red flower with white flower and if I get a pink flower, I know that this gene is a codominant gene which is showing its heterozygosity. So a codominant marker lets you know the heterozygosity of the plant, but a dominant marker cannot detect the heterozygosity. It can only detect the presence, whether the gene is present or not, but a codominant marker will tell you whether the gene is present in homozygous condition or in heterozygous condition. So dominant markers cannot differentiate between homozygote and heterozygote, right? So codominant marker can differentiate between homozygote and heterozygote, that I was telling about. So molecular markers can be linked, gene based or functional. For example, the deletion that I was giving the example just before, that deletion may be on the causal gene itself, or it may be in a very nearby location, so closely placed that the deleted part and the gene, they do not recombine. So this kind of a marker are known as the linked marker, and sometimes we get even better marker which are functional marker. Means I know that due to a point mutation, the CAG codon has changed to TAG codon, and I know that due to this TAG the gene is not going to be transcribed and translated properly. So the function and the marker is linked, then I call it a functional marker. So linked marker means marker is present at nearby position of the candidate gene. There is chance of recombination between candidate gene and the marker. I will be taking another class on your 2-point and 3-point test cross, where I will be elaborating this matter that why a linked marker is not sometimes preferred, because there is a chance of recombination between linked marker and the candidate gene. And gene-based markers are located on the candidate gene itself. Candidate gene means the gene which is directly governing the trait, right? And functional means it is obviously a gene-based marker located on the candidate gene, and the sequence variation is validated with the gene function. Means I know that why the gene function is changed due to this sequence variation. So this becomes a functional marker. So this functional marker is having a very high breeding value, and it's very robust in nature. So molecular marker may arise due to insertion, deletion or substitution mutations, as I have already mentioned. They are reliable, robust and free from environmental effect that I was discussing, that if a particular trait or a particular disease require a particular environment to occur, but if you are using a molecular marker, even in a non-favorable environment, you can say whether this particular genotype is going to be resistant or susceptible to a particular disease. So they facilitate selection of multiple traits simultaneously. Suppose you are screening for root knot resistance as well as red fruit color as well as a pericarp thick tomato. So all these traits can really be selected, multiple traits can be simultaneously selected if you use molecular markers, and they greatly improve the selection efficiency. As I have told that the codominant functional markers, they can differentiate between homozygote and heterozygote lines. So you can have an early generation fixation of lines when you select the plants which are already homozygous for a particular gene or a particular mutant allele. You know that in the filial generation also you are going to get the phenotype of the mutant only, not the dominant one, not the wild type one. Means your population size can really be made reduced. So it helps in recovery of the genome in back cross breeding program, MABC. I will be taking another lecture on MABC where I will be discussing that how the molecular markers helps in background genome recovery when you go for marker assisted back cross breeding, and useful in preparation of genome maps and identification of QTLs, identification of candidate genes through fine mapping and map based cloning of genes. So now let's come to the basic 10 types of the molecular markers. The first one is RAPD, sometimes pronounced as rapid. This was one of the oldest form of molecular markers, DNA based markers that we used. It is full form is randomly amplified polymorphic DNA. Here you need not to know any genomic information of any crop, and you can use this marker because this is a PCR based marker where a very short, around 10 nucleotide long oligonucleotide primer is used. Remember, in RAPD we use only a single primer, okay? So this primer, as you can see here, this primer can have attachment in different positions of the genome. Sometimes they attach in the same direction and sometimes they attach in the opposite direction in two opposite strands. So due to the nature of polymerase chain reaction where we have the exponential growth in amplification, only the regions where the primer is attached in a tandemly inverse repeated manner, they will give some banding patterns, right? So you can say here we have to use a very low annealing temperature in case of RAPD because the length of the primer is very short. The same primer may randomly bind in complementary strands to give the amplification. No genome sequence information is needed, but the problem is that it's very poorly reproducible. The lab to lab, batch to batch variation in the banding pattern while we are working with RAPD is very high. So you can see here it's parent 1, parent 2 and the F1. You can see that RAPD cannot actually discriminate the homozygous and heterozygous plants, but they can give you the indication of the presence. For example, the parent 1 had the 1, 2, 3, 4, 5th band present and parent 2 lacked the 5th band, but in F1 plants all the bands are present. So this is dominant in nature. It might be converted to a reproducible scar marker. I will be coming to the scar later on. Next coming is the RFLP marker. It's a little bit more cumbersome but a little bit more reproducible. Here we use restriction enzymes as well as random amplification. So this is a kind of technique where in RFLP we cut the genomic DNA, and depending upon the variation in the restriction site sizes, positions, you can get different size of fragments, And if you use a radio-labeled probe which is complementary to the region of your interest, then you can see you can have differential banding pattern in the autobiography which can act actually as an RFLP. So this is restriction digestion of genomic DNA is followed by transfer in southern blot method. The region of interest is used as a denatured radio-label probe and hybridization takes place. The problem is a very cumbersome technique involving the radio labeling of probes and radioactive elements handling, and high quality and quantity of genomic DNA is needed for restriction sites. Previously, when we didn't have so much advancement in the case of molecular markers, RFLP was a technique that used to give us several linkage maps and several position of genes. Next comes the technique AFLP, which is actually a mixture of RAPD and RFLP. You can say it is amplified fragment length polymorphism. It is having much more reproducibility than the previous two. So it's a hybrid technique. Generally it uses two restriction enzymes, a hexacutter and a tetracutter. So a tetracutter becomes a frequent cutter. It produces more cuts in the genome. Here in the pictorial diagram you can see that it digests the genome in different, and we get different fragments of the genome. Then what we do, following digestion, specific adapters are ligated to the fragments. So these adapter sequences are known to us. So now having this adapter joined, now we can have amplification of these adapter ligated fragments. It's done basically in two steps. In the first step, you have a selective primer where one arbitrary nucleotide is added in the 3' end, and after that we go for again a selective amplification with radio labeled selective primer where up to 3 arbitrary nucleotides are incorporated. So this reduces the number of bands that you want to actually generate. Otherwise there will be so much bands that will be very much difficult to score the gel, but here the reproducibility is much more high and you get relatively less amount of bands, and this is a technique AFLP which helped previously to have idea of several genetic regulations and development of molecular markers. Next comes the one of the most popular marker that is used, that is SSR, simple sequence repeat. That, as I have mentioned, that the genome is basically highly composed of repetitive sequences. Now what happens, the number of repeats is generally highly conserved, but when DNA polymerase work in these regions, then due to the repeat, DNA polymerase seldom makes some mistake in replicating. So the number of repeats, they vary within the same species in different varieties. So this gives the rise of SSR markers. The two sides of these repeat motifs are highly conserved, and depending this conserved sequence, we design two primers and we amplify. So generally these SSRs are single loci markers. They generally amplify a single band of the particular genomic fragment. In some of the cases it can give rise to multiple bands also. So tandem repeat of sequences of up to 1 to 6 base pair, and they are repeated 5 to 50 times, right? So the number of repeats is highly variable. The flanking regions of the repeats are highly conserved, on the basis of which we design the primers. Then two specific primers are used to amplify a single locus. Here you can see we are using two primers, long primers. We can have high annealing temperature, so there will be very less chance of non-specific binding of the primers. So it's highly reproducible and it is perfectly codominant in nature. Here you can see in parent 1 we had a bigger band, in parent 2 we had a smaller band, and in F1 plants we had both the bands. So presence of both bands proves the heterozygosity of the plants, and parent 1 and parent 2, they are producing only single band. So it's a perfect codominant marker. So very small amount of isolated genomic DNA is needed, but here you need to know the genomic information, because to design the two specific primers you need to know the sequence of the conserved region. Next comes the ISSR technique, which believes on the occurrence of these SSR motifs, and generally we apply this ISSR technique for those crops where we expect some SSRs are present, but we don't know the genomic information of the crop. So in case of ISSR, what we do, we target the amplification of the region between two simple sequence repeats. So for example, suppose there is one repeat GA, 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 and there is another repeat CT, 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 CT. So if you have a primer GAN ending with arbitrary nucleotide, then what will happen? If the primer anneals in two complementary strands, then it will be amplifying a region. And it is a multi-loci primer. Unlike SSR, which amplifies a single loci, ISSR will be binding in all the repetitive regions present in the genome and will be giving you a very nice fingerprint. So sometimes ISSR is used for fingerprinting, means particularly in tissue culture to test the genetic fidelity of the plantlets, the ISSR techniques can be used. So very small amount of DNA again is needed. Reproducibility again is an issue in case of ISSR because it's a kind of RAPD. You can see no sequence information is needed and it is suitable for DNA fingerprinting. Next I come to SCAR, means I have already discussed the RAPD technique, and you can see that suppose there is one band you can see which is coming in a RAPD but not present in other parents. So what we do, we simply cut this gel band and then we clone it, and after cloning it you know a longer sequence of this band, then you can design some specific primers which are around 20 nucleotide long, so that now your PCR reaction will not hit in all the genome but only in the particular site from where this RAPD was coming. So depending upon the nature of this appearance or disappearance of the RAPD band, you can develop a dominant scar, or you can come across, if you are lucky enough, you can come up with a co-dominant scar. So this is the example how a RAPD marker can actually be converted to a SCAR marker. So due to short random oligonucleotides, RAPD is highly non-reproducible. So in the SCAR technique, we overcome that very thing, right? So we design longer primers around 20 nucleotide. Depending upon the nature of polymorphism, the newly developed primers may have dominant or co-dominant type amplification. In case of several crops, a number of SCAR markers, linked SCAR markers have been developed which have been proved to be very much useful, particularly in disease resistance breeding. So now this is one of the most powerful markers, that is SNP. It is a single nucleotide polymorphism. Remember previously all the markers that we were talking about, they gave a size polymorphism, difference in band size, but it may be that the two fragments are having the same size, but only a single nucleotide sequence is changed between them. So this gives rise to the SNP marker system, single nucleotide polymorphism. It's most abundant in the genome, and SNP can be substitution, addition or deletion, can occur in both genic and non-genic region, can occur in coding DNA sequence. SNP not changing amino acid called synonymous SNP, because due to this change the amino acid is not going to be changed. Non-synonymous SNPs leads to missense or nonsense mutation. It's most preferred marker, it's codominant in nature, but low cost detection of SNP is very much challenging. Means you go for resequencing in NGS approaches, you identify a lot of SNPs. For example, in variety 1, the sequence is AGG, AGT, GT, and in variety 2, the sequence in AGA, AGT, CT. So this GC gives you an SNP, right? So validation of SNP, then you go for detection of it by fluorescence based method, allele specific PCR, and other high throughput methods. So I will be discussing one of these methods where how we can actually identify an SNP even in low cost techniques, right? So the first technique is CAPS, a cleaved amplified polymorphic sequence. This is a sequence, suppose you have a SNP T2A, right? In one variety it is T and in the second variety it is A. Now luckily this T is a part of a restriction site. Suppose the site is GGA-TCC, and in the second variety it becomes GGA-ACC. So due to this T2A change, the restriction site is also abolished. GGA-TCC is the restriction site for BAMH1 restriction enzyme, but GGA-ACC is not the restriction site for BAMH1. So if we amplify this region through the conserved primers, and if we digest it, then in parent 1, you look, this 400 base pair band will be cut into two pieces, one 250 base pair fragment and one 150 base pair fragment in parent 1. But in parent 2, it will not be cut into two parts. And in F1 plants, which are TA in nature, you will have all the three bands. So this is cleaved amplified polymorphic sequence marker, CAPS marker, which is also a codominant marker. This is a little bit cumbersome because you have to do the PCR, then you will have to digest the PCR product, and then you have to run the gel. And second, if you are not lucky enough, this sequence change may not be a part of a restriction site. So what happens if the sequence change is not a part of the restriction site? Can we make it a part of the restriction site, right? So this technique becomes DCAPS, or derived cleaved amplified polymorphic sequence. So here you can see the sequence is CGA-TCC, which has been changed to CGA-ACC. So here what we do, we simply incorporate one or two mismatches in the primer, so that in one case we get a restriction site recovered, and in the second case the restriction site is not recovered. For example, if we make a primer, forward primer like capital GG, then it becomes, after amplification, GGA-TCC, and here it becomes GGA-ACC, because the nucleotide that you incorporate in the primer is always present in your amplified product. So this restriction site will be present in one amplified product from a variety, but not present in the other one. So now if you digest it, then the length of the primer, around 20 to 25 base pair, that will be removed from the amplicon. So it will give a smaller amplicon after digestion, just like in parent 1. You can see there was a 200 base pair amplicon, and after digestion 20 base pair is removed, you get a 180 base pair band. But in case of parent 2, no restriction site is recovered, so it is not digested and remains 200 base pair. So F1 plants are heterozygous, they are giving rise to both the bands. So this is a very powerful technique through which an SNP can actually be scored in a gel based manner. Without sequencing, you can predict whether it is a T allele or it's a allele, right? So only one amplicon can be restriction digested and cleaved. That gives the nature that it is codominant in nature. It's a perfect marker on the basis of functional SNP. There are several DCAPS marker that has been identified and developed for identifying functional SNPs in the genes. Now coming to another gel based technique, that is T-ARMS, which does not require any restriction digestion to find an SNP. The full form is tetraprimer amplification refractory mutation system. Here actually we use 4 primers, right? So the 2 primers are out forward and out reverse. They will give the band in all varieties. Now if the SNP is G or A, then we have one inner primer that ends in G and a second inner primer which ends in A. The G primer is a forward primer and A primer is a reverse primer. So deliberately the position of GA SNP is kept in such a manner that when in G forward and out reverse will give you a band, the size of that band will be different from the size of the band generated by out forward and in A reverse. So there is a very simplified way that I have told you all these things. So increase the specificity of the primers, a deliberate mismatch is incorporated in the -2 position of the primer. So look at this allele. When out forward and out reverse will give you a common band of 400 base pair in all the cases, right? So if there is G, if the gene is containing G, then only in G forward and out reverse primer can work. In A reverse cannot work because there is no A. So the G allele will be giving you a 400 base pair band, common band, and a 300 base pair G allele as depicted in parent 1. But in case of parent 2, which does not have the G allele but the A allele, here in G forward primer will not work, but in A reverse primer will work with the out forward primer to give you a band of around 200 base pair. So in one parental line you have a 300 plus 200 banding pattern, and in the 400 plus 200 300 banding pattern, and the second you will be having 400 plus 200 banding pattern. So only by looking at the banding pattern you can say that whether the G allele present or the A allele is present. So you are detecting the SNP in a gel based manner, and what will happen in case of F1 plants, all the 3 bands will be visible. So this technique, as simple it looks, but it is a cumbersome one. The optimization part, the primer concentrations, are several optimizations are needed to have this kind of clean picture, right? So it's a perfect marker based on the basis of functional SNP, but the optimization part is difficult. So that's all for today's lecture. I give you the 2 suggested readings. One is the Biotechnology Expanding Horizons by B.D. Singh, and the second one is Introduction to Plant Biotechnology. And I request you to please give your comments in the comment box that you will be seeing. And if you have any question, please feel free to contact me. My contact details will be available in the university website also. Thank you very much.